Well, I think the mechanism of action of both of these products is very important to understand. So for Inebilizumab, this is a B-cell depleting monoclonal antibody. Many NMO patients are receiving care from MS specialists. MS specialists have a lot of experience with B-cell depleting monoclonal antibodies because they've used rituximab in the past and now ocrelizumab for treatment of multiple sclerosis. Inebilizumab falls in the same class of drugs that targets B cells and is administered with the same frequency of dosing. So I think there's going to be a certain familiarity with the class of compounds that are B-cell depleting monoclonal antibodies that will be appealing to many clinicians. In contrast, eculizumab is a terminal complement inhibitor. Complement is used as part of the body's innate immune system to prevent bacterial infections, and there are additional risks associated with terminal complement inhibition that one has to be aware of, including the potential for serious bacterial infections, including bacterial meningitis. Now, with eculizumab, we do immunize patients beforehand, and that does provide quite a bit of relief. But I think the challenge there is long-term use of the product in NMO patients, and how that is going to be with respect to long-term safety, and how one's going to manage patients who develop fever, and what kind of workup one wants to do. With the B-cell depleting antibody, we don't have those same concerns. The B-cell depletion may be associated with some increased risk of infection, but we really didn't see that very much in the NMO trial. And we haven't really seen that very much with ocrelizumab or rituximab either, although there may be some association with herpes virus reactivation with ocrelizumab. It remains to be seen whether we're going to see something similar like that with inebilizumab. I think the labels mentioned about infusion reactions is a class effect, because we know that with monoclonal antibodies, there can be infusion reactions. We didn't really see those in this trial. And in fact, the inebilizumab treated patients tended to have fewer infusion-related reactions than the placebo-treated patients, which is very different from ocrelizumab. And I think that has to do with how the drug depletes B cells. Now, I don't know that for sure, haven't proven that. But with ocrelizumab, there is some complement-mediated lysis of B cells, so very fast depletion of B cells. Same with rituximab. In contrast, with inebilizumab, this is all through ADCC and ADCP-mediated cell-based depletion of these B cells. Takes a little bit longer time, but I think as a consequence of that, you have fewer infusion-related reactions. That's speculation on my part, can't prove it, but we see far fewer infusion-related reactions with inebilizumab. So the language in the label, I think, just is there as a class warning, and because this is a relatively small study, 231 patients. So I think the FDA is showing appropriate caution in including a broader statement that they have learned from other monoclonal antibodies. I would say there is a real concern, though, with inebilizumab with respect to depletion of gamma globulins. So because inebilizumab is targeting a broader range of B cells than, say, for example, ocrelizumab, there are plasmablasts that are depleted, as well as pro-B cells that are depleted with inebilizumab. As a consequence of that, we are seeing with long-term exposure to the drug some depletion of gamma globulins, and some patients do develop hypogammaglobulinemia. And so as part of the label, we do have to check an IgG level before and then periodically during treatment, and especially in patients who have low gamma globulins and develop an infection, would be something of concern. So it is something we're going to be keeping track of as we roll this product into clinical practice. I'm certainly going to be checking IgG levels on my patients in advance and making sure that I'm not treating somebody with this product who already has hypogammaglobulinemia to begin with. That was part of the exclusion criteria for the clinical trial.